I'd like to get started with the next set of talks, so if everybody could take their seats. All right, our next speaker has become a major figure in the world of PANS and PANDAS. Her name is Madeleine Cunningham from the University of Oklahoma. It's a pleasure to have you here in St. Louis. Thanks, Madeleine.
Thank you so much, Dr. Slager. And I also want to take this opportunity to thank the 5 parents who have been so, actually 6 parents, the group that has been so wonderful in their courage and their motivation and stamina to actually put together this meeting, because we're all here because of them. Thank you so much. Today I'm supposed to be talking to you about autoimmunity and PANS and PANDAS, and we'll give you a little bit of introduction to myself. I've spent 33 years working on acute rheumatic fever and myocarditis and quite a number of autoimmune heart diseases. So Dr. Sweeto called me about 15 years ago, and she said, can you help us work on this neurologic disorder? And of course, Sydenham, Korea was part of the rheumatic fever complex. So of course I said yes. And this is 15 or 16 years later. I'll present you with what we have done over that period of time. I have a number of disclosures, as most of you probably know, that I'm involved with Molecular Labs, who is here. But I've been funded for years, for about 27, 28 years, from the National Heart, Lung, and Blood Institute, and also from the National Institute of Mental Health, and a number of other agencies that are listed on the slide. I don't know what happened to my, maybe I probably don't have this. Yeah, I'm going to take care of that right now. I'm going to talk to you first because so much of my work has been about the group A streptococcus for 33 years. As you know, there are many diseases it can cause: pharyngitis, impetigo, scarlet fever, septicemia, and then there are other more serious diseases that you've heard about, the quote flesh-eating bacteria causing cellulitis and necrotizing fasciitis. But acute glomerulonephritis and rheumatic fever are the 2 post-streptococcal, quote, autoimmune sequelae of the suppurative infection. The autoimmune manifestations of rheumatic fever include polymigrating arthritis, which is the most common manifestation, carditis, which of course is the most serious, and then the Sydenham chorea, which is the CNS manifestation. There are a couple of other skin manifestations, erythema marginatum and subcutaneous nodules. And we studied this disease so long that, this almost looks like a rundown of the systems that you see in systemic lupus erythematosus. And we do know that some of the autoantibodies produced in rheumatic fever, we produced years ago anti-idiotypic antibodies against those antibodies, and show that they're present only in lupus and Sjogren's, but not in other types of autoimmune diseases, so that it has a link with systemic lupus. Just to show you the guidelines for, these are the Jones criteria, primarily that you have 2 major manifestations and a minor manifestation. You can see what they are on the slide, and most of you are probably familiar with this, but there has been a revision, and I want to make you aware of that has just come out in circulation this year, and I have a copy of it if any of you would like to see it. It's with me, and it revises about the heart condition and the relationship to data that you would collect by Doppler. So it's a good resource for you and diagnosis of acute rheumatic fever. As Dr. Sweeto pointed out, molecular mimicry is an important mechanism between the group A streptococcus and the heart, brain, and joints, where the organisms incite an antibody response in the human host that actually then attacks the heart, joint, or brain. You can actually have chorea and carditis in the same individual. And Dr. Gene Stollerman, my mentor, actually used to say that if you had carditis with chorea, the carditis would be worse. But this, of course, there are many stories from the pre-antibiotic era. So the associated diseases, behavior and movement disorders have been discussed today already by Dr. Sweeto and Dr. Mink. Our approach to the study of these diseases, and has been for a very long time, is to study humans by translational research, and so we have produced human monoclonal antibodies, taking B cells from people and making them live forever, so that we have a reagent that actually never ends, that we don't run out of sera, because we can make these monoclonal antibodies from people. And so we made them and looked at the brain and streptococcal antigens, and then went on to study the pathogenesis of the disease. The mimicry in Sydenham chorea we found using monoclonal antibodies from these people reacted with the group A streptococcal carbohydrate in acetyl beta-D-glucosamine epitope, and then also with lysoganglioside and tubulin brain antigens. And just to give you a little smattering of the data so you don't feel like you didn't hear any of it, the lysoganglioside you see in red, it took a very small amount to inhibit these monoclonal antibodies from a patient with Sydenham chorea that I saw in the hospital there at the University of Oklahoma. And so we identified that antigen, but many others you can see would not inhibit it at all by many of the gangliosides that identify Guillain-Barré syndrome. So it's very specific. And if we looked at Sydenham chorea sera, Dr. Sweeto provided a set of patients where they were acute sera and then convalescent sera. And the convalescent sera is very important because that is when the patients begin to recover. And you can see in the red that the antibodies to the lysoganglioside are elevated in the acute, but are on the decrease in the convalescent sera. We began to study these antibodies in PANDAS patients compared to Sydenham chorea, and also looked at Tourette syndrome, OCD, ADHD, and normals. These sera provided by Jim Leckman and by Sue Sweeto to us. And you can see that the anti-lysoganglioside antibodies are significantly elevated in PANDAS, SC, and the Tourette's group that was provided to us that did not have OCD. They only had tics. The Sydenham chorea and PANDAS cerebrospinal fluid, it's very important to realize that CSF, or cerebrospinal fluid, is a great resource for studying and understanding these diseases. Taking these, this CSF fluid, we demonstrated in the red shaded areas on the right. You can see how the Sydenham and PANDAS antibodies from the cerebrospinal fluid probably bound nerve tracts in the brain, but these are not seen in the control samples where we used antibodies from people that did not have these diseases. Anti-tubulin antibodies, which do stain the brain, were present in PANDAS, Sydenham chorea, and also in tics, but not in the OCD, ADHD, or controls. So we ask ourselves, about this time, we're thinking, well, how would these antibodies work? We know that people with Sydenham chorea don't die. They don't seem to have permanent damage, and they do, after maybe years of time, get better. So we studied these antibodies against live cells and found, as you can see, dark purple blobs of antibody that are on the surface of this human neuronal cell line. And so we found that the antibodies, including our monoclonals, which are much easier to study than the serum, actually signal through a calcium calmodulin-dependent protein kinase 2 in neuronal cells. And you can see here the monoclonal antibody 24, one of the 3 that we had produced. The other 2 make great controls because they are not so signaling of the CAM kinase. You can see the activity of this enzyme over basal rate in a human neuronal cell line. And then, of course, you would expect that the donor who gave us that monoclonal antibody, that their serum would reflect what that antibody demonstrated, and of course it did. Their serum in red, their convalescent serum months later when they were well did not have the signaling antibody. So this would speak to the idea that perhaps antibodies that signal may play a role in Sydenham chorea and potentially in PANDAS. So Dr. Sweeto came to the rescue again and provided sera from 5 patients with Sydenham chorea that she had seen and treated at the NIMH. And she had 3 sera that were convalescent when they had recovered, and you can see that these antibodies are reduced. And then the mechanism by which these antibodies may work is that they actually bind to human neuronal cells and then induce a dopamine release so that excess dopamine is released from these neuronal cells and then potentially could play a role in disease. We looked at PANDAS and Sydenham chorea together along with the non-PANDAS group. And you can see that the CAM kinase activity, the signaling activity of the Sydenham chorea sera versus the PANDAS sera versus the non-PANDAS and control sera show a stair-step signaling cascade with the SC higher than the PANDAS, and then of course the non-PANDAS at a particular level of signaling. The antibody then, we would predict, binds to the neuronal cell, inducing tyrosine hydroxylase and CAM kinase activity. And in fact, when we put the antibodies intrathecally into the brains of rats, you can see on the right the pink staining indicating that the antibody induced tyrosine hydroxylase. So this is the pathway to producing dopamine in the brain. So we also wanted to ask if there was evidence for a role potentially of the dopamine receptors. If we potentially have excess dopamine, are the receptors maybe playing a role? And the hypothesis that we put forth were that dopamine receptors present on the surface of neuronal cells react with and respond to antibodies as if they were neurotransmitters such as dopamine. And so we expressed in a transgenic animal the human antibody from the Sydenham chorea case. And this antibody actually reacted with, I don't know if is this a pointer? Oh, it is. The antibody actually was produced in serum. This is not staining of tissue. This is where the antibody actually traveled from the serum into the brain of these transgenic animals. And so we stained for the presence of the antibody after we broke the blood-brain barrier. And so the human antibody travels into the brain in these animals, and these neurons are actually in what we believe is the substantia nigra based on the anatomy of the tissue section. And the red staining is for tyrosine hydroxylase, indicating that the antibodies targeted dopaminergic neurons in the brain of a transgenic animal that we converted into a mouse that had the antibody from Sydenham chorea. It was an IgG. We converted it to an IgG. It took a long time, about 9 years, to create this animal. So that was very interesting and actually indicated that the antibodies could penetrate the blood-brain barrier and that we were right, that they could penetrate dopaminergic neurons in the brain. So the antibodies were potentially, could they also react with dopamine receptors D1 and D2? And yes, in fact, we were able to get data indicating that in PANDAS and in Sydenham chorea that the antibodies against the D2 receptor were present in people with choreiform movements. And you do in the PANDAS, acute onset PANDAS with strep, you can see choreiform movements also in those children, but not to the extent of Sydenham chorea. Dr. Sweeto can address that if you have questions about it, the actual types of movements. So the monoclonal antibody, then, we wanted to find out if it actually would signal D2 on a cell. And so we did have cells that were transfected with the dopamine D2 receptor, and it's an inhibitory receptor, so you can see how it actually decreases the cyclic AMP. And it's just as good as dopamine. You can see where dopamine signals. And the dose response was beautiful with increasing concentrations of the monoclonal antibody. So just to move ahead maybe a little more quickly, we also found that peptides would inhibit the antibody and that a flag would track with the D2 receptor. This was just all the proof that in fact the antibodies were reacting with the D2 receptor, and that the Sydenham chorea sera also signals just as the D2 receptor, just as the monoclonal, and the PANDAS sera also signals this receptor just as good as dopamine. So they do act like a neurotransmitter. They're very unusual antibodies, and they act in nanogram amounts. If I tried to test these on a Western blot, you would barely see them because they're in such a small quantity, but they're very powerful. So what about the D1 receptor? Then we also looked, and antibodies to the D1 receptor were also present, particularly in PANDAS and then in Sydenham chorea, but not as significant because there was a large range, but not in any of the others except for OCD. There was a significant level. This is, I don't have time really to go into more detail about this antibody, but this is a monoclonal antibody obtained from the PANDAS patients that were in the recent IVIG trial that Dr. Sweeto is getting ready to talk to you about. And briefly, this antibody did signal the D1 receptor expressed on cells, and it also has another type of mechanism. It actually makes the receptor 100 times more sensitive to dopamine. So you can imagine if patients are making too much dopamine, and then they're making their receptors more sensitive to the dopamine. And there could be even another scenario. What if they don't degrade their dopamine in a way that's normal? Then those are three mechanisms. That's what I'm supposed to be talking about to you, is how it could happen. So genetically it could happen by having a gene that maybe expressed a variant and you wouldn't degrade your dopamine. Producing too much dopamine with antibodies like the one from Sydenham chorea and PANDAS, or having an antibody to D1 that sensitized the receptor and made it much more sensitive to the activity of dopamine. So those three ways are important in potential mechanisms of how the antibodies could play a role. So we have a Lewis rat model. Dr. Sweeto mentioned animal models. We have a Lewis rat model. Dr. Hornig has a mouse model. And in our streptococcal induced model, we immunized with group A streptococcal antigen and determined that it led to animals that could not hold a food pellet, also that they could not walk a narrow beam like normal mice, and that they groomed excessively, unlike normal mice. And Dr. Hornig had animals that she had treated with streptococcal antigen, and she found that these animals just excessively jumped and jumped and jumped. I don't have the video for that, but important for you to know that it's not only one but two models, and she passively transferred this behavior with antibody. In our model, we also were able to treat with haloperidol and paroxetine, and actually reduce the symptoms. I know there are a lot of little dots. Here's the becoming more normal, and then of course in the grooming becoming more normal with the use of paroxetine or haloperidol. And these antibodies did deposit in the thalamus, in the striatum, and the cortex, and not in the cerebellum or the hippocampus. So IgG antibodies from streptococcal immunized rats induced CAM kinase and were absorbed with anti-IgG, which we always try to do. We prove that it is IgG that's causing the signaling by an absorption with anti-IgG beads, and these antibodies in the animal sera reacted with the dopamine receptors in Western blots. So again, we still ask the question, is Sydenham chorea a potential path to understanding neuropsychiatric disorders? And we tried to reproduce our data that from work that we had done with Dr. Sweeto in Sydenham chorea. Dr. Hilla Pazzi had 22 patients with Sydenham chorea. And we studied her patients and found that the D1 antibody in the group overall comes up first, and the D2 antibody comes up second. The D1 coincides with the more early neuropsychiatric symptoms, whereas the D2 antibody coincides more with the movement, and that is what we see. I showed you data we had from studies of groups of sera, and in fact, the D1 had some significance with OCD, and the D2 more with the chorea, because D2 is not seen oftentimes in patients with some of these diseases. So the distribution of D1 and D2 is shown here from Dr. Pazzi's study. They were both significant. And she also found that the ratio of D1 to D2 antibody correlated with the neuropsychiatric symptoms and the USCRS score, as shown here. And so the model of autoantibody effects in the brain, we do believe that binding of high avidity cross-reactive antineuronal antibodies to lysoganglioside and dopamine receptors may cause alterations in dopamine transmission. I'm going to go back over those three mechanisms again. One mechanism being the antibody causing too much dopamine production. That's the anti-D2. The anti-D1 monoclonal antibody that we made from PANDAS actually binds to the D1 receptor and sensitizes it, making it 100 times more sensitive to dopamine. And then the possibility of having a genetic variation where dopamine would not be degraded. Those are three important mechanisms that may be active in this disease, or these types of diseases. We have some new evidence that I'm going to quickly show you, and then I'm almost finished. Evidence for autoantibodies in more chronic tics and OCD that would have greater than 1 year of symptoms. There are two new studies, one directly from our laboratory based on samples that we collected from patients over about a two-year period. And we studied 261 patients that we had confirmed streptococcal-associated infections. And the antineuronal antibodies are in this heterogeneous group of youth and young adults with chronic tics and obsessive compulsive disorder. And our study suggested a significant correlation of streptococcal-associated tics and OCD with, right here, elevated anti-D1 and anti-lysoganglioside antibodies in serum, but even more specifically and significantly associated with the signaling of the CAM-K2 in the human neuronal cells. Also, Dr. Harvey Singer headed up a study from Johns Hopkins where the neuronal antibody biomarkers in Sydenham chorea were studied in his patients. It was a longitudinal study of a small number of patients where we found in some of them increased anti-D1 and anti-lysoganglioside antibodies, But the single most important marker was the CAM kinase in these children. And these antibodies we found didn't go up and down. They were already chronically elevated in these children because many of them had been sick for even past a year. And so if the antibody's already present, then it's possible that any type of infection could lead to breaking the blood-brain barrier and entrance of the antibodies into the brain. And so finally, just to show what we found with this particular group, that tics and OCD together were significantly associated with the presence or reporting of strep. The concept of autoantibodies and autoimmune diseases has long been the history that groups of autoantibodies against different antigens can be associated with a particular type of autoimmune disease. In PANDAS, PANS, tics, OCD, or some other neuropsychiatric diseases that may be associated with inflammation, autoantibody groups against certain neuronal antigens and receptors are or may be a characteristic feature. And then the two-step process of the disease is that we expect that immune responses against an infectious pathogen or an environmental agent would cause antibody that would react in the brain, and then opening of the blood-brain barrier due to infection or stress would allow antibodies to penetrate the brain. And I also would like to take this opportunity to tell you that these diseases probably are associated with immune complexes, because rheumatic fever has long been known to be associated with a high rate of immune complexes. And just in like lupus, which we know already from our data that rheumatic fever is linked to lupus by their immune responses, that it also has immune complexes. And in both diseases, I suspect they are not cleared properly, and so they hang around for a very long time, causing vasculitis and damage to the vessels in the brain. And that not only are you getting this type of response that I've been talking about, but you're also causing other damage, including maybe even fibrosis to the vessels in the brain. So I think with that ending, I'd like to thank my whole laboratory and all the people that have helped make this possible. From Yale University, Dr. Leckman, and Tel Aviv, the animal model, Dr. Joel's group, Dr. Ben-Pazi, Dr. Sweeto, certainly in her group, and Dr. Rebecca Homer, Dr. Fallon at Columbia. We are studying Lyme disease with him in adults, and Dr. Singer for his patience putting up with me, and then Dr. Janicki and Dr. Geller, who were so supportive of the study that we did of almost 1,000 patients, and then our statistician, and then Dr. Kaplan, who was a strep infectious disease doctor in Minnesota. Thank you very much.